Photosynthesis and Cellular Respiration by Grace Morris. Photosynthesis and cellular respiration are interconnected processes that sustain life. Photosynthesis occurs in the chloroplast of plant cells, capturing light energy to produce glucose and oxygen. Cellular respiration occurs in the mitochondria, breaking down glucose into ATP. Let's examine how energy flows through these reactions. The light-dependent reaction begins when sunlight strikes photosystem II in the thylakoid membrane, exciting two electrons. These electrons travel through the electron transport chain. To replace the lost electrons, a water molecule is split, producing two hydrogen, two electrons, and oxygen. Oxygen is released as a waste product. As electrons move through the electron transport chain, their energy pumps hydrogen protons into the thylakoid space, creating a proton gradient. These electrons reach photosystem I, where light excites them again. NADP+ then combines with two electrons and hydrogen to form NADPH. Meanwhile, ATP synthase allows hydrogen ions to pass back into the stroma, generating ATP from ADP and inorganic phosphate. The Calvin cycle uses ATP and NADPH from the light-dependent reactions to convert carbon dioxide into glucose. In carbon fixation, 3 CO2 molecules combine with 3 molecules of RUBP with the help of enzyme Rubisco, forming 6 molecules of 3-PGA. In the reduction phase, ATP and NADPH convert 3-PGA into 6 molecules of G3P. 1 G3P exits the cycle to help form glucose, while the remaining 5 G3P molecules regenerate RUBP using ATP. To make 1 glucose molecule, the cycle must turn twice, using a total of 6 carbon dioxide, 18 ATP, and 12 NADPH. Glycolysis occurs in the cytoplasm and breaks down 1 glucose molecule into 2 pyruvate molecules. The process requires 2 ATP and produces 4 ATP, resulting in a net gain of 2 ATP. Additionally, 2 NAD+ molecules capture electrons, forming 2 NADH. If oxygen is unavailable, cells undergo fermentation instead of aerobic respiration. In alcoholic fermentation used by yeast and some bacteria, pyruvate is converted into ethanol and carbon dioxide, also regenerating NAD+. Though fermentation only produces 2 ATP per glucose, it allows cells to continue generating energy when oxygen is absent. In lactic acid fermentation, pyruvate is converted into lactic acid, regenerating NAD+ to sustain glycolysis. This process occurs in muscle cells during intense exercise. When oxygen is present, pyruvate enters the mitochondria. In pyruvate oxidation, each pyruvate loses a carbon, SCO2, and binds with coenzyme A to form acetyl-CoA. This process also produces 2 NADH molecules, which will be used in the electron transport chain. The Krebs cycle takes place in the mitochondrial matrix. Acetyl-CoA joins with oxaloacetate to form citrate, a 6-carbon molecule. Citrate breaks down, releasing 2 CO2 and producing 3 NADH, 1 FADH2, and 1 ATP per turn. Since the cycle runs twice per glucose, the total yield is 6 NADH, 2 FADH2, 2 ATP, and 4 CO2. NADH and FADH2 go onto the electron transfer chain to make more ATP. The electron transport chain takes place in the inner mitochondrial membrane. Electrons from the 10 NADH and 2 FADH2 pass through the electron transport chain, pumping H+ into the intermembrane space. Oxygen acts as the final electron acceptor, forming 6 H2O. As H+ ions pass through ATP synthase, 34 ATP are generated. This completes the cycle. Photosynthesis captures energy, while cellular respiration releases it at 38 ATP per glucose.